There are over 20,000 genes in the human genome and only 23 pairs of chromosomes for them to fit. This means that there are going to be a lot of genes that end up on the same chromosome. Now, if we only focus on, let's say, 2 genes that each have 2 alleles apiece, and they're on the same chromosome, and we look at the allelic combinations we could get after meiosis in our gametes, we'd find that instead of getting 4, we'd only end up with 2. And this would appear to break the rule of independent assortment that says all these alleles can segregate independently and we get all these neat combinations. Well, if the point of sex is to increase this genetic variability, then we need a way around this problem. And our germ cells have a really cool trick to do this and make sure that we can mix and match all of our alleles. It's time we move back to visual representations of our chromosomes because this really cool trick called chromosome recombination is a bit more complicated than what we can do with our deck of playing cards. Again, we're not going to show all 46 chromosomes or all 23 pairs. I'm just going to show a representative set of chromosomes here. Here we have 1, 2, 3, 4, 5, 6, 7, 8 pairs of chromosomes. Of course, each pair has 1 chromosome from mom in pink here and 1 chromosome from dad in blue. Now, chromosome recombination is a very special kind of event. Now, in the exercise we just looked at, we had an individual heterozygous for lactase persistence and dwarfism. But we hypothetically imagined that these alleles were on the same chromosome, chromosome 2. And because of this, after meiosis occurred, we only got 2 possible allelic combinations because these alleles are on the same chromosomes. So the law of segregation still applies, but independent assortment, where alleles from one gene assort independently from alleles of another gene, doesn't seem to hold true. Instead, these alleles appear linked. What a special trick does, called chromosome recombination, is it allows regions of a chromosome to actually swap information. What this means is that physically, literally, these chromosomes are touching each other, right? One of these chromosomes came from mom, one of these chromosomes came from dad. They're going to get so close that they're going to connect at regions along the chromosomes, and where they connect, they're going to exchange pieces of information so that what happens is a bit of mom and dad's chromosomes get mixed up. They get swapped. So now this one linear chromosome that used to be the whole chromosome that you got from mom is now a chromosome that starts as a piece you got from mom, then includes a bunch of sequence you got from dad, and then more sequence you got from mom. So when you pass these chromosomes on to your children, you're not just passing down mom's chromosome that she gave you or dad's chromosome that he gave you. You're passing down a mix of your mom and dad's chromosomes. Now think about what this means for the allelic compositions. Let's just imagine for a second that the placement of our lactase and dwarfism alleles are here on the chromosomes. And we know already that if recombination doesn't occur, we only get 2 possible allelic combinations here. But if recombination does occur, look what happens. Now, whereas the dominant and recessive alleles were on the same chromosome here, you have the 2 dominant alleles for each trait on the same chromosome and the 2 recessive alleles together, so that when segregation occurs, we get the 2 missing combinations that we didn't have before. So just to make sure you're with me again, I want you to tell me how many gamete genotypes are possible in this case, even when genes are on the same chromosome but far enough apart for chromosome recombination to occur.